Hi, it's Wendy at the Barrington Library. Today we're going to be making edible DNA. So this will be fun. It involves a little bit of candy, so hope you have fun doing it. I'm going to turn my camera down. In your kit, you'll be getting a bag with some gumdrops. There should be 4 different colors in there. You'll get some toothpicks and a couple of Twizzlers. So I have already opened up my Twizzler. And the next thing to get ready for my kit would be to open these up and separate them by color. So I'm going to go ahead and do that. While I'm doing it, I'll explain a little bit about DNA. So I don't know if you know, there are 37 trillion, over 37 trillion, not million or billion, but trillion cells in the human body. And within those cells, in the nucleus inside the cell, there are important molecules called DNA. DNA is the molecules that hold your unique genetic code. And DNA is short for deoxyribonucleic acid. Ooh, that's a hard one. But that holds all the instructions for each of the cells in our bodies. What they're for, how they grow, mature, when they die, things like that. It has all the instructions of what the cells are going to do. There we go. And DNA contains 4 natural types of nitrogen bases that make up 4 nucleotides within the DNA. Adenine, thymine, cytosine, and guanine. Or it could be adenine, thymine, cytosine, and guanine. Sorry about that. That's called ATCG. Adenine is always paired with thymine. So in this case, our adenine is green and our thymine is white. So these are the 2 that will be paired together. And then always, cytosine and guanine will be paired together. And the cytosine is the black and the guanine is the orange. So when we're making up our pairs to go down our DNA molecule, we want to make sure that we're keeping these 2 colors paired and these 2 colors paired. Because in our DNA, those 2 types of nucleotides are always together and these 2 types are always together. So we unwrapped our licorice and we separated our gumdrops, and I have my toothpick, so let's get started. Remember, green is always with white. So I'm going to go ahead and put a white on and a green on. It's a little sticky. I'm going to stick it near the top of my licorice. And then this one's on the other side. I'm going to push it in. All right, so I can feel those little pokes on the end there, but I didn't stick them way too far out through, but that's okay. So there's one set, 1 pair. Now I can choose to do a pair of the next colors, or I can do 2 more in a row of this one. This is where you get to alternate what you want to make for patterns or not, because it's always mixed up and different within our own DNA also. I'm going to go ahead and do another one of these, but what I'm going to do, instead of putting them on this way, I'm just going to flip them that way. A little wild here, huh? Okay, what I'm going to do is probably poke in that side and then do this side later on. So let's go ahead and get this color. There. We are making up these pairs. And imagine a very microscopic, teeny tiny molecule inside your cells. Do you know cells are so small? They have to use very super high-powered microscopes to be able to see our cells. And then within that, well, our cells are like 1,000 times smaller than a millimeter. And I don't know if you can see on this ruler, we have inches on one side and millimeters and centimeters on the other. And this 1 centimeter is broken up into 10 millimeters, those teeny weeny white lines. So a cell, you could fit 1,000 of them in between those lines. So that's how small cells are. And then the DNA molecules inside that are even smaller. So they need very high-powered equipment in the laboratories for scientists to see this. I'm going to go ahead and put the same colors on again. And no, I'm not going to flip them. I'm just going to put them beneath each other. And then the next one, I'm going to add this one, and right beneath it, I think you can fit probably 6 or 7 on here. So we will try. We will see how it goes. One more after this, maybe. There we go. Yeah, I'll do one more. So as you see, I have not mixed up my colors. I've kept the pairs with each other. Now I'm going to connect this side. Push it through till I feel it on that side. I can feel it with my thumb come through. And this is connecting all of our nucleotides. There we go. Looks like a little ladder, doesn't it? Now, just imagine, now that we're done, we'll twist it a little bit. Just imagine that this is what your DNA looks like under a microscope. Pretty cool, huh? And it holds together pretty well. And then it looks like a ladder. And that's what it is. There. Oh, and look at this. I have leftover candy. Nothing saying that you can't eat the leftover candy. Or even your DNA double helix if you want. Hope you had fun, guys. Have a great day. Bye-bye.